Yeah. So can someone unmute? Can someone unmute and let me know if my screen is visible and if I'm audible?
Yes.
Okay, thank you. So, good evening, everyone. Today is the first session of discussing questions related to the biochemistry course. So, you guys must have enrolled in the biochemistry course. That is, the course instructor is Professor Subramaniam, K. Subramaniam. And in these live sessions, every week we'll have one live session. And in these live sessions, basically, we'll be discussing some questions that came as assignments previous year, and we'll discuss those questions and we'll discuss the concepts behind those questions, so it's easier for us to solve similar kind of questions in the future. So my name is Aishi and I'm currently a third year PhD student in the Department of Chemistry, IIT Bombay, and we'll start with today's session. We'll discuss some of the problems and we'll try to discuss the concepts. If anyone has a doubt, maybe you can unmute and let me know, or you can just type in the chat box and I'll check the chat box, and then maybe we can have a discussion regarding the same. Okay, so let's start with the first question for today. So the first question for today is, which of the given scientists were involved in the discovery of the double helix structure of DNA? Okay, and it's mentioned that more than one option is correct. So does anybody want to try? Which option or options do you think will be correct? Second and fourth. Okay, anybody else? Okay, so I get where everyone is coming from. Watson and Crick got Nobel Prize for DNA double helix. I think someone wrote something in the chat box. Can you unmute and let me know if you have a different answer? Okay. Okay, so obviously Watson and Crick are famous for the DNA double helix model, but Maurice Wilkins was also part of the Nobel Prize, by the way, which was awarded Nobel Prize in Physiology or Medicine, which was awarded in 1962 to James Watson, Francis Crick, and Maurice Wilkins for the discovery of the molecular structure of DNA, which helped obviously solve one of the most important of all biological riddles. So this structure was revolutionary because from a long time scientists were trying to figure out the structure of DNA, and they were not able to. So these three people got the Nobel Prize for the structure. However, Rosalind Franklin was also a very important figure, and even though unfortunately she was not awarded the Nobel Prize, it was basically her X-ray data, X-ray diffraction data, that actually led Watson and Crick towards the Nobel Prize. So without her data, nothing would have been possible, and people actually came to know about it quite later, and I think she died because of cancer, and at that point of time, Nobel Prize was not given posthumously. So she actually lost the Nobel Prize recognition, and that is why she's one of her contemporary titles was she was called the Dark Lady of DNA, because it was her data basically that actually led to one of the biggest discoveries of a generation, and she was not even awarded the Nobel Prize for the same. So we'll discuss a little bit about the DNA double helix structure and the contributions of all the scientists. So the answer will be actually all four. Watson and Crick are the most famous for double helix structure. Maurice Wilkins also shared the Nobel Prize, and the basic data that gave rise to the DNA double helix model was actually from Rosalind Franklin's X-ray diffraction data. So if we talk about the basically nucleic acids, so nucleic acids are obviously the main function is that it's the genetic material of all cells, be it bacterial cells or eukaryotic cells. Nucleic acids are obviously the genetic material which carry on genetic information to the offsprings as well. And in obviously it stores information, that is the most important parameter, and in the form of genes, and they're obviously the blueprint for building proteins. So any kind, whatever the genetic information is there, they are being the proteins are being encoded by that genetic information. So if there is any kind of mutation, your proteins will also probably be the mutated form. It might be misfolded or it might not fold properly. So obviously DNA, RNA proteins, we know about the central dogma, DNA to RNA transcription and RNA to proteins translation. And this is the DNA is a double helix structure which ultimately gives rise to RNA and then proteins. Okay, and it transfers information definitely because the same genetic material is being passed down to the next generation, to their offspring, which is why it transfers information. It's a blueprint for the new cells. And so whenever there is daughter cells being formed from the mother cell, obviously genetic component, the DNA is during cell division, the DNA is being replicated and each pair is being passed off to the daughter cells. So it should be identical to the parent cell. So obviously it's a blueprint for the new cells and it's a blueprint for the next generation. Okay. So moving on, for examples, like we have nucleic acids, we'll have the RNA of the ribonucleic acid. Okay. And ribonucleic acid is not exactly the genetic element. It is mostly present in the form of a single helix. However, we are more important in DNA, deoxyribonucleic acid, which is much more stable, and this is a genetic element that is known to be passed out from mother cell to the daughter cells, basically to the offsprings. Okay. So the DNA double helix, the structure is obviously the monomers are the nucleotides. So obviously, basically it's a polymer. So the monomers will be the nucleotides. The different nucleotides that form that are the monomers, and ultimately the nucleotides form a chain, ultimately giving rise to the polymers, and ultimately that is the DNA double helix structure. So the nucleotides are every nucleotide can be different. Can be categorized into three parts basically. The nitrogen base, if you can see the structure over here, we have the nitrogen base over here, we have this is the nitrogen base, we have the pentose sugar. So nitrogen base can be either adenine, thymine, guanine or cytosine, and for RNA it can be uracil as well. Okay, so in DNA, in place of uracil we have thymine, and in RNA we have uracil. And this nitrogenous base that is a prominent element of nucleotides. And then we have the pentose sugar, which is basically it's a 5-carbon sugar. And then we have, which is ribose in RNA and deoxyribose in DNA. So this is one of the features that actually make DNA much more stable than RNA, and that is why it has been, evolution has basically chosen it to be the genetic element and not RNA. So this is the difference between the two structures, deoxyribose in DNA and ribose in RNA. And then we have phosphate. For the phosphate group is over here, which basically gives DNA a negative charge. So obviously the nucleic acids are charged molecules because of the phosphate group over here. So nucleic of the DNA will be negatively charged. So types of nucleotides, if we can see, there will be purines or pyrimidines. Adenine and guanine are the two different types of purines, while cytosine, thymine and uracil in RNA are the three different types of pyrimidines. Okay, and they'll have, so in the purines we have double ring N base, whereas in case of pyrimidine we'll have a single ring N base. That is quite self-explanatory from the images as well. And cytosine, thymine and uracil are the pyrimidines. Okay, so basically what is the backbone of the nucleic acid? The sugar to pentose sugar to phosphate bond, this is phosphodiester bond, is the backbone, basically forms the backbone of the nucleic acid. And then the new base is added to the sugar of the previous base, and the polymer obviously grows in one direction, and whereas the N bases hang off the sugar phosphate backbone. Okay, So obviously, dangling bases. So is this important? Why are the bases hanging off the sugar phosphate backbone? So we'll talk about this also. Obviously, directionality of DNA is also very important. Okay, so in a double helix model, which was ultimately deciphered by Watson Crick from the data of Rosalind Franklin, the double helix, the two different strands of the DNA are actually anti-parallel to each other. Okay, so if we look at this structure, so we can refer to the 3' and the 5' ends of the DNA. That refers to the last trailing carbon. So this will be the 5' end of the DNA, and this will be the 3' end of the DNA structure. Okay. Now, obviously, there are hydrogen bonds between the different strands of a DNA double helix. So this is one strand of the DNA double helix, this is the other strand, and these are anti-parallel to each other, and they are linked with the help of hydrogen bonds. Okay, so we need to remember that adenine and thymine, they have actually two hydrogen bonds between them, whereas GC, that is guanine and cytosine, they are linked with 3 hydrogen bonds. Okay, and if you can see the structure, the two different strands are anti-parallel to each other as well. So these are the covalent phosphodiester bonds, and in the middle the two strands are linked to each other with the help of hydrogen bonds. Okay, AT and GC. So in case of DNA molecule, we have talked about the double helix structure, and so what is the groove length between one? So basically the groove length will be 3.4 nanometer. So these H bonds between the bases join these two strands. Again, AT will have only 2 hydrogen bonds, whereas GC will have 3 hydrogen bonds. Okay. Now hydrogen bonds, why is this important? Okay. If we think about adenosine triphosphate, which is the basis of energy in our system, ATP, it's a modified nucleotide, and it ultimately gets hydrolyzed to AMP and inorganic phosphates. At first ADP plus 1 inorganic phosphate, and then AMP, adenosine monophosphate, and 2 inorganic phosphate groups. Okay. So this is another important factor, if you can see. So these are the three, these are the phosphate groups, and this is it's basically a modified nucleotide, and that basically holds the energy center. Basically, if we are talking about glycolysis or Krebs cycle, whatever we're talking about, and in mitochondria, which we call the powerhouse of the cell, basically generates ATP. So adenosine triphosphate is basically a modified nucleotide. Talking about the difference between DNA and RNA, obviously DNA structure has deoxyribose as a sugar. It's a double strand, double helix, whereas RNA is single helix, and RNA has ribose as a sugar. Okay, DNA has thymine as one of the nucleotides, but RNA will have uracil. Okay, so these are the differences between main difference between DNA and RNA. It will have uracil in place of thymine. Okay, now. At first, there was Erwin Chargaff, who studied the amounts of nitrogenous bases in DNA, and he came up with a formula which basically help us, if we know the amount of cytosine in a DNA structure, we'll be able to decipher the amount of guanine in the structure. So basically, he put forward some formulas based on the studies that talked about the amounts of nitrogenous bases in DNA, and we get the base pair rule. Then came Rosalind Franklin, who actually performed X-ray diffraction studies to get pattern from the structure of DNA, and she is the one who's responsible for the molecular structure. So Watson and Crick have been working on this, deciphering the structure of DNA for a long time, but they were not able to figure out exactly how the two double strands are linked with each other. So it was Rosalind Franklin's X-ray diffraction data which was given to Watson and Crick by Maurice Wilkins without the knowledge of Franklin. Her data was passed on, and ultimately Watson and Crick published the model and paper based on Rosalind Franklin's data without her knowledge, and did not even give her credit for the same thing. So James Watson and Franklin, no doubt they were also working on similar thing, but it was impossible without Franklin's data. Without Franklin's X-ray diffraction data, nobody would have been able to come up with the DNA double helix model, which is why it is very important that we remember the contributions of Franklin. And even though she was deprived of the Nobel Prize, she was actually the most deserving candidate for the Nobel Prize that was given for DNA double helix. Okay, so what is the role of genetic material? A genetic material must carry out two jobs. It should duplicate itself, obviously, and control the development of the rest of the cell in a specific way. Okay, we have talked about DNA, and obviously the information necessary to sustain and perpetuate life is found within this molecule, and it is genetic material that is passed from one generation to the next. And before the mid 1800s, it was unthinkable for the leading scientists and philosophers that a chemical molecule could store this much information to actually build a human. Correct? So cells transfer their genetic element to the daughter cells, and this is how exactly the offsprings get the genetic element from their parents. So it was actually unthinkable before a time period that a chemical molecule can hold this much information. And in 1859, Charles Darwin, when he published The Origin of Species, it was expressed that living things appear to be designed but may be the result of natural selection. And Darwin showed that living creatures evolve over several generations through a series of small changes, which is basically the concept of evolution. And obviously Darwin's ideas were finally supported because of the studies by Gregor Mendel as well. Like factors determine the characteristics of a living thing. The living thing will express there will be a dominant factor and a recessive factor, and the genes will get passed to a later generation with a child taking genes from both the parents. But still the mystery was how is this information stored, and which molecule is basically responsible for this kind of information. So the search of genetic material was already going on when German scientist Friedrich Mischer actually isolated the chemicals found in the nucleus, and he found that those consisted proteins and nucleic acids. Okay, and while he found these nucleic acids interesting and obviously spent a lot of time studying their chemical composition, he actually wasn't the only one who was studying the same. There were other people who believed that proteins were more likely to be the chemicals involved in inheritance because of the immense variability. So we know that proteins are really variable. DNA is a stable molecule, and DNA is transferred from generation to the next generation. Proteins are much more variable. So many people were of the opinion that proteins are the chemical molecules responsible for heredity, responsible for transferring information from one generation to the next. Okay, and proteins were made up of 20 different building blocks or amino acids, as opposed to the mere 4 building blocks of nucleic acids. We have only 4 nucleotides, right? ATGC in DNA. So as opposed to that, we have 20 different amino acids. So people were of the opinion that proteins probably were the chemicals involved in inheritance. Okay, and then Frederick Griffith led to the discovery of a transforming principle. He saw that pneumonia obviously infected mice, and mice developed pneumonia and they were dead in a few time span. But there were 2 types of bacteria: S bacteria with a smooth coat that caused pneumonia, and R bacteria with a rough coat that did not cause pneumonia. And this coat type is associated with virulence. He found out that this coat type is associated with the virulence of the bacteria. So then he did a genetic material experiment. So that's how he came up with the transforming principle. Sorry, one sec. Yeah. So do you guys need anything? I feel very distracted. Sorry. Yeah, so the search for genetic material continued when Frederick Griffith came up with the transforming principle, and he saw that the SS strain of the Pseudomonas was able to cause infection, after which the mouse dies. But the R strain, which was a living R strain without the smooth coat, even after infection, the mouse was still healthy and it did not die even after some time. So he came up with the virulent and non-virulent strain. So the living S strain was the virulent one and the living R strain was the non-virulent one. Okay. So it was seen that when you heat kill the S strain, it could not cause the infection anymore. So the virulent S strain was causing the infection, but after heat killing the S strain, it was not able to cause the infection anymore. But if we mix the heat killed S strain with the living R strain, now R strain was non-virulent, it was not causing any infection anyhow. But if we mix the heat killed S strain with the living R strain and then infected the mouse dies. This should not be, ideally the mouse should have been healthy because the S strain is heat killed, which we know that keeps the mouse healthy, and living R strain is anyway not able to cause any kind of infection. But when we put an infection, when we put a mixture of both these, heat killed S strain and the living R strain, and we infect the mouse died. And he could actually isolate living cells of S strain from the dead mouse. So Griffith's experiment actually identified the transforming principle. He found out that there are two different kinds of bacterial colonies, the rough virulent strain R and the smooth virulent strain S. And the heat killed smooth virulent was also strain S. But when we mix the rough strain R and the heat killed virulent smooth strain, it actually killed the mouse. So there was something that was getting transformed in this case, because individually both of these were not able to cause infection. But when they were mixed together, something happened, infection happened, and the mouse died. Okay, and after that, live strain S bacteria in blood sample could be isolated from the dead mouse. So what happened in the fourth experiment? The R cells were harmless and it had been transformed by bacterial from the dead S cells, and the descendants of the transformed cells were also pathogenic. So the question was, what was it that was getting transformed or transferred? So the extracts of heat-killed S bacterial cells contained protein, RNA and DNA. Now they were trying to figure out which among these substances were essential for this transformation. And how did they figure out which substance was essential for transformation? Later, the other key players were Oswald Avery, who actually showed that DNA is a unit of inheritance. Okay, and his work actually inspired Watson Crick to actually go in this journey for seeking DNA structure. Okay, so Oswald Avery was an American bacteriologist who proved that DNA is the molecule that was getting transferred. It was the one that was genetic material. Okay, so now they tried to discover which was the molecules which one was the molecule was responsible for this genetic element transferring by the process of elimination. So they treated that with either proteases to destroy the protein, with RNAs to destroy the RNA, or even DNAs to destroy the DNA. And they found that the transformation was exclusively due to DNA. So when DNAs was used to destroy the DNA, nothing could be seen. So from this experiment, they could find out that transformation was exclusively due to the DNA. So Avery found that cell extracts treated with protein digesting enzymes could still transform bacteria. But cell extracts treated with DNA digesting enzymes lost their transforming ability. So he concluded that DNA and not protein transforms bacteria. Okay, and then his colleagues were still studying the bacteria which causes pneumonia and discovered that bacteria contain nucleic acid and that DNA is the chemical which carries the genes. And despite the conclusive results of Avery's experiments, the theory of nucleic acids being the genetic material was still not a popular one. But experiments performed with viruses also showed that nucleic acids were the genetic material, and this confirmed Avery's work. And after that came the experiments by Hershey Chase. So Alfred Hershey and Martha Chase in 1952, they used a bacteriophage, which is basically a virus that infects bacteria. And they used this to prove that DNA was the hereditary material, and that the bacteriophage was the ideal organism for settling the debate between protein and DNA. So what are viruses? Viruses are basically organized associations of macromolecules and nucleic acid contained within a protective shell of protein units. So virus in itself is not alive because it does not have proteins like proteins that can help in their structure and function. It does not have those of their own. So virus in itself is not alive. It is not made up out of a cell. So alone it cannot do anything. It needs a host. It will enter a host and then hijack their entire machinery and replicate its own nucleic acid and basically hijack the entire machinery of the host. So those are viruses. So they use bacteriophage, which is basically a virus that infects bacteria. So the DNA, the genetic element, is basically inside this protein core, and we have a hollow sheath. Okay. Now the question was, what infects the bacteria, the protein or the DNA? which one is getting inserted into the bacteria when infection occurs. So via Hershey-Chase experiment, they actually labeled the protein capsule with sulfur, and they labeled the DNA with phosphorus, and then they performed the infection. Okay, and then they saw that there was no sulfur detected in the cells, but sulfur was detected in the supernatant. That means since sulfur was labeling the protein capsule, no sulfur was detected in the cells. That is, no protein was getting inserted from the bacteriophage into the cells. However, they could detect phosphorus inside the cells and no phosphorus in the supernatant. So phosphorus was labeling the nucleic acids, the DNA code, and the DNA is getting inserted, and phosphorus was detected in the cells. So this was another revolutionary experiment Hershey-Chase in 1952. You can imagine the time period when this was performed. And they actually discovered that DNA was what was causing the infection, and the search for genetic material was still continued, and basically not the protein code labeled with sulfur, but the DNA material labeled with phosphorus was getting inserted into the cells. Okay, and Hershey and Chase basically they showed that protein does not infect the bacterium. Okay, so if you can see, the virus protein was labeled with sulfur, but sulfur remained outside the cells. But the DNA was labeled with phosphorus, and the phosphorus was seen inside the cell. So that is how they show that DNA infects the bacterium, not the protein component. And so these were the classic experiments that were performed for the evidence Griffith transforming principle. Then we have the Hershey-Chase, which finally gave evidence that DNA was necessary for the infection by the bacteriophage. And then there were other supporting evidence, which is basically we knew that DNA volume doubles before cells divide, because obviously in the S phase of the cell division, DNA replicates itself. And then we had the ratio of nucleotides from Shargov's tool, Erwin Shargov. Okay, so who discovered that base pairing it's basically a complementary relationship of nucleic acids that provided one of the key steps in developing a structural model for DNA. So his Shargov's tools were basically helpful, because now Francis and Crick had a model to look forward to, because he found the complementary relationships of nucleic acids. Okay, he discovered that pairing rules of DNA letters, noticing that A will match to adenine, will match with thymine, and guanine will match with cytosine. Okay, so later came the work of Watson and Crick. James Watson and Crick were ultimately working on the double helix, and after solving the double helix, they all went to work on viruses and RNA. And he also apparently helped in the Human Genome Project. Francis Crick was also involved in discovering the structure of DNA. He went on to crack the genetic code that translates DNA into protein. So these were basically the double helix structure was discovered by Francis Crick, James Watson, and Maurice Wilkins using the X-ray diffraction. So they were actually discovered that the structure of DNA double helix, or interlocking pair of spirals joined by pairs of molecules. Okay, and the X-ray diffraction data was actually of so James Watson and Crick basically they shared an office, and they were trying to work on it, and they were inclined to follow the methods of Pauling, who had designed a helical structure by building a model consistent with the X-ray patterns from fibrous proteins. So like proteins, DNA was built from similar units like adenine, thymine, guanine, and cytosine, and so it seemed likely that DNA will also have a helical structure. Okay, so but the published X-ray patterns of DNA were not very clear. Okay, so he attended a DNA colloquium in 1951, where Rosalind Franklin described her results. Okay, now Rosalind Franklin was an English chemist, and she had very good X-ray photographs of DNA, and basically these were the crucial contributions to the solution of the structure of DNA. Okay, so her images of DNA disclosed without her knowledge was given by Maurice Wilkins to Watson and Crick, and finally they went on to so they basically used her data with other evidence to describe the structure of DNA. This was the X-ray diffraction photo of DNA image produced by Rosalind Franklin. Okay, And obviously they took into account Shargow's ratios also, that the ratios of AT and GC were statistically equal for a wide variety of DNAs. And then Crick became convinced that base pairing was the key to the structure. Okay, and so Watson again visited King's, and Wilkins showed him a DNA X-ray pattern taken by Franklin and showing clear helical characteristics. Okay, so from this, what they deduced from Franklin's X-ray data was that it was a double helix. It had a uniform width of 2 nanometer and bases was stacked 0.34 nanometer apart, and which was also pushed forward with because of Shargow's rules, which basically talked about complementarity, where adenine pairs with thymine and cytosine can pair with guanine. So what they came up with their own was that bases face inward and phosphates and sugars outward, and there was hydrogen bonding, and they also hinted at the semi-conservative model of replication. Okay, and they found that A plus T and G plus C could be bonded together to form pairs with very similar shapes. So on this basis, a model was built consistent with both Franklin's symmetric data and Shargow's results, and they finally published it in Nature, accompanied by ones from the Wilkins and the Franklin groups. And finally, it's a very good Nature paper, and the last sentence of the Nature paper is that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So they also hinted at the semi-conservative mode of replication of DNA. Basically how the DNA strands they separate, and each strand gives rise to 2 different daughter strands. So this is basically an image of Watson and Crick with their DNA model. So obviously Crick, Watson and Wilkins, they won the Nobel Prize for Medicine or Physiology in 1962, and Rosalind Franklin did brilliant work, but she received little recognition because by that time she was not given recognition, and finally she actually died in 1958, so she could not be recognized in the Nobel Award because at that point Nobel Awards were not given posthumously. So this is the Nature paper. It's a very fascinating read, so you can basically read it in your free time. Okay, so we have talked about the vocabulary of DNA and the DNA facts, and how DNA can be how Rosalind Franklin and Maurice Wilkins took DNA X-ray photos that were essential to the discovery of double helix, and basically Rosalind Franklin was not given the recognition, and later people came to know, and we as students who are studying science, we should know the contributions of Rosalind Franklin, without whom the structure of DNA could never have been deciphered. Okay, so the DNA strand is like this only. Each nucleotide bonds to the next one to form a strand, and the 2 strands then twist around a central axis to form a DNA double helix. Okay, and the sides of the ladder alternate between a phosphate and a sugar deoxyribose, and these rungs are held together by the hydrogen bonds. If you can see, there are 2 hydrogen bonds between adenine and thymine, and 3 hydrogen bonds between guanine and cytosine. Okay, so adenine can bond only with thymine, AT, and cytosine can bond only with guanine. This is the base pair rule that was pretty clear from Shargow's rules. Okay, so there are 4 bases, and that can have endless combinations. So they're just like the letters of the alphabet can combine to make an infinite number of bonds. So the 2 strands are said to be complementary. That means if you have GAATAC on one side, you will have an antiparallel strand complementary to this one on the other strand. Then about the replication, the Francis Crick paper in Nature, they actually hinted at semi-conservative mode of replication. So the process by which the DNA makes a copy of itself. So in DNA replication, enzymes will work to unwind and separate the double helix and add complementary nucleotides to the exposed strands. So you can see this was the original DNA strand. They separated at one point, and this was the old DNA strand. This is also the old DNA strand, and new DNA strands is being made complementary to these two separated old DNA strands. So that makes DNA replication is semi-conservative. So when it makes a copy, one half of the old strand is always kept in the new strand, and this helps reduce the number of copy errors. Okay, so this is the DNA strand shown in the form of a ladder. They separate, and new strands are being made complementary to the old strands. So 2 new strands are created, and each contain half of the original strand. So DNA helicases are there. Basically, they break the H bonds linking bases. Obviously, DNA polymerases are there which move along each of the strands adding nucleotides according to the base pairing rules, and the result is two exact copies of the original DNA. Each new double helix is composed of one original strand, the blue one over here, and one new strand, the red one over here. So this is the semi-conservative mode of replication. Okay. Now translation. DNA is in the nucleus obviously. To make proteins, DNA must get its instructions to the ribosomes to make proteins. So for transport, to transport its instructions, basically it uses messenger RNA or mRNA. So by the process of transcription, RNA is transcribed from a DNA template, and then RNA processing happens. So in eukaryotes, the RNA transcript is spliced and modified to produce mRNA, which moves from the nucleus to the cytoplasm. And in the mRNA leaves the nucleus and attaches to the ribosome, and then each amino acid attaches to its proper tRNA with the help of a specific enzyme and ATP. So a succession of ATPs add their amino acids to the polypeptide chains as the mRNA moves through the ribosome, and one codon at a time. So this is how the DNA is transported from the nucleus outside in the form of mRNA to form proteins. In case of RNA, we have already talked that it consists of one strand of nucleotides, has a ribose, not deoxyribose, and it has a uracil as a nitrogenous base, not thymine. So if we talk about numbers, each cell has about 3 meters of DNA, and the average human cell has average human body has 300 trillion cells. So the average human has enough DNA to go from the earth to the sun more than 400 times. Okay, and DNA has a diameter of around this. Okay, so you can figure out how folded and how condensed the DNA might be, because we have such a huge length of DNA, but everything is fit inside those cells. Okay, so we have talked about Frederick Mesher and his experiments. We have talked about how early researchers were still doubtful about DNA and doubted if it was the hereditary material and if it was proteins. Then we have talked about Frederick Griffith and his experiments in using S-coat bacteria and R-coat bacteria in mice, and we have talked about how that something gets transformed, which is basically a transforming principle. We have talked about Oswald Avery and how he isolated proteins, carbohydrates, everything, and saw that only nucleic acids caused a change. So that was Avery's work. Then we obviously talk about Erwin Chargaff, who studied DNA itself, providing clues about its structure. We discovered that there are always equal amounts of bases adenine and thymine and equal amounts of cytosine and guanine. And he also proposed with his base pair rule that these bases pair with one another in some way. Then we have Wilkins and Franklin, who worked with X-ray crystallography to find more clues about the structure of DNA. And Franklin's data basically helped solve the DNA double helix. We were also talked about James Watson and Francis Crick, who were also working on discovering the structure of DNA. They applied Chargaff's rules, and Watson was not entirely convinced of the helical structure that Franklin had suggested. And basically, after they consulted Wilkins and after they saw Franklin's data, they actually came up with the first DNA model. Okay, and that DNA is made up of 4 bases. RNA also has 4 bases, but has uracil instead of thymine. Okay, so across the DNA ladder, A will always pair with T and C will always pair with G because of the number of hydrogen bonds. So GC will form 3 hydrogen bonds and AT will form 2 hydrogen bonds. And the DNA ladder can form a spherical or helical structure with the 2 sides being held together with hydrogen bonds. Okay, and we have talked about DNA replication, that before cells divide, they must double their DNA so that each cell can get identical copies of the DNA strands. And we have talked about the semi-conservative mode of replication. The hydrogen bonds break to unzip the DNA strand, and enzymes guide free nucleotides to the exposed single strand and match the nucleosides based on complementarity. Okay, so in summary, we have talked about how DNA was discovered and which people should be given credit for the same, and we have talked about all the components of DNA and how if there was the helical structure, of if there was any kind of hydrogen bonding between the structures, but if there were other pairing structures, for example, there's something called the Hochstein pairs, and they were still in a dilemma if adenine actually paired with guanine, and if adenine actually paired with thymine, and if guanine actually paired with cytosine. So there were also a lot of problem, and finally the the tautomer issue is also there, but finally based on the Shargav's rules, which came before that by Edwin Shargav and obviously Rosalind Franklin's data, people came forward with the DNA structure. Okay, so we'll move to we have talked about the extracrystallography structure as well and how the data looked like. So this will be the extracrystallography structure of a helical fiber, and this was exactly what people got from Rosalind Franklin's data. Okay, so we have also talked about the structural details. So coming back to the question that we started with, all 4 of the scientists were involved in the DNA double helix, not only Watson and Crick, even Morris, Wilkins and Rosalind Franklin's X-ray diffraction data were important. So the answer to that question will be all the 4 options will be correct. One second. Yes, so it will not only be Watson and Crick, all the 4 answers would be all 4 scientists were involved in the DNA double helix structure deciphering. Moving on to the next question for today. The salts of which of the following ions contribute to the hardness of water? Sorry, is anyone's mic on? Can you please? Yeah. Okay. Yeah. Okay. Anybody else? Okay. So yes, obviously the answer will be magnesium and calcium. Can you unmute? Mute your mics, please. I am getting a lot of noise over here. 1 second. Is someone coming? Yeah. So yes, magnesium and calcium. So water which does not produce any kind of lather with soap is termed as hard water. Okay, and this hardness is usually expressed in terms of these 2 divalent cations like calcium and magnesium salts like bicarbonate, carbonate, sulfate, and chloride. So hard water is formed due to the presence of minerals like calcium and magnesium. They are not removed or separated by sedimentation or filtration. So when hard water reacts with soap, which is the sodium salt of stearic acid or palmitic acid, it gives the curdy precipitate. So these are the equations basically. And in the above reaction, hard water reacts with sodium salt of stearic acid to form calcium stearate or magnesium stearate, which being insoluble and separate out without producing lather. Okay. Now, there can be different types of hardness. Obviously, there can be temporary hardness and permanent hardness as well. So, temporary hardness of water is caused by calcium and magnesium bicarbonate, and this can be removed by simply boiling of water. And due to the boiling, bicarbonate will be converted into carbonate, which will form an insoluble precipitate. Okay, and temporary hardness can actually be removed by adding hydrated lime to precipitate the insoluble carbonate over here. Permanent hardness, on the other hand, is caused by the presence of soluble salts of calcium and magnesium other than bicarbonate, not exactly bicarbonate, such as chloride and sulfate. So, permanent hardness cannot be removed by boiling of water or hydrated lime. It can be eliminated by water softening techniques like lime soda process, zeolite, ion exchange resin, reverse osmosis, etc. Okay. So, the unit in which hardness is usually expressed is known as the degree of hardness, and degree of hardness is expressed in terms of calcium carbonate equivalent. 1 second. Calcium carbonate equivalent. Okay, so this is how the degree of hardness is shown in equivalence of calcium carbonate. Okay, it can be shown by parts per million, which is the number of equivalent parts CaCO3 present per million part of water by weight. It can be shown number of milligram of CaCO3 present in 1 liter of water. So this is how the units of hardness can be shown. Okay. So what are the techniques for water softening? Zeolite process is there, which is a 3-dimensional silicate, and the chemical formula is hydrated sodium aluminum silicate. It is a huge formula over here, and then basically capable of exchanging ions with sodium ions. So it's capable of exchanging hardness producing ions present in water, and this process is also called as permutate process. Okay, over here then we have it can be regenerated also. So the exhausted zeolite can be regenerated by treating with brine solution, which is 10% NaCl solution. Okay. We can go for ion exchange resin process as well. So in this process, cations and anions are completely removed by passing impure water into two different columns. The first column contains sulfuric acid resin with acidic group, and this column is known as cation exchange resin because it exchanges only cations. Whereas the second column contains resin with basic groups, and it is known as anion exchange resin because it exchanges anions. Okay. So the removal of H plus ions from the first column and the OH minus from the second column will react to form the water. And even in this case, in regeneration, when both the columns are exhausted, the first and second column can be treated with dilute H2SO4 or HCl and aqueous NaOH. So they have to be regenerated. Okay, then we have the lime soda process as well. So in this process, hydrated lime and soda ash can be used to remove the hardness from water. Okay, and it can be used, obviously we know that hydrated lime is used to remove the temporary hardness of water because it can react with the calcium and magnesium salts to form insoluble precipitate of calcium carbonate and magnesium hydroxide. Okay, and hydrated lime can also be used to remove permanent hardness from water. It will react with magnesium sulfate and magnesium chloride to form insoluble precipitate of the magnesium hydroxide. Okay. We have reverse osmosis also. The minimum excess pressure that has to be applied on the solution to prevent the entry of the solvent molecule into solution to semi-permeable membrane is known as osmotic pressure. Okay, and so in this case, if a pressure higher than the osmotic pressure is applied to the solution, the solvent will flow reverse in a reverse osmosis. So you can see the image over here. This is the semi-permeable membrane. This is a higher concentration of solutes, lower concentration of solutes. So there will be flow from here to here. But in case of reverse, if there is a pressure applied over here, there will be reverse osmosis. The higher concentration solution to low concentration solution, and this process is known as reverse osmosis. And this process can also be used in purification of seawater. For this purpose, seawater is delivered under pressure through the semi-permeable membrane, where water permeates the minute pores of the membrane and is delivered as purified water. Okay, 1 second. Purified water. Okay, so obviously what are the advantages of reverse osmosis? No maintenance, it removes colloidal silica and is gaining ground for converting seawater into drinking water. Okay, so these were the different going back to the question that we started with, the salts of which of the following ions contribute to hardness of water? It will be magnesium and calcium. Okay, so moving on to the next question for today. 1 second. Which molecule is involved in transfer of genetic information? We have already discussed about this. Yeah, correct. Nucleic acid is involved in transfer of genetic information. So obviously, but there's obviously importance given to lipids, carbohydrates, and especially proteins because they are the macromolecules that are also able to do a lot of other things, and these are the basically 3 of the 4 classes of lives organic molecules, and these are polymers. All of these are polymers. Okay, so the diversity of polymers is very fascinating. So each cell has thousands of different macromolecules, and most of these macromolecules can act like monomers to form polymers. So carbohydrates will include sugars, and the polymers, basically they are polymers of sugars. The simplest carbohydrates will be the monosaccharides or simple sugars, and carbohydrate macromolecules are polysaccharides, where the polymers are composed of many sugar building blocks. Okay. And the monosaccharides will have a molecular formula very simple like CH2O, and they'll have a molecular formula in multiples of CH2O, like for glucose, for example, this is H12O6. This is the most common monosaccharide, and monosaccharides can be classified by the location of the carbonyl group like aldose or ketose and the number of carbons in the carbon skeleton. If you see, these are trioses because they have 3 carbon sugars, glyceraldehyde and dihydroxyacetone. So the functional group in this case is an aldehyde, and the functional group in this case it's a ketone, dihydroxyacetone. It's a ketone group. The CO is a ketone group, and here we have an aldehyde. But these are triose, 3-carbon sugars. For example, the 5-carbon sugars that we have, pentoses, we have ribose and ribulose. Hexose, obviously we have glucose. So glucose, we have glucose, and we have fructose, which is a ketone sugar, and we have glucose, which is basically an aldehyde. So though often they are drawn as linear skeletons, in aqueous solutions they actually form rings. And monosaccharides are important because they can serve as major fuel for cells and a raw material for building the molecules. 1 second. Yeah. So a disaccharide is formed when a dehydration reaction will join these two monosaccharides. Okay, and this covalent bond will be called the glycosidic linkage. Okay, so we can see over here the glucose and glucose will get together and they'll form a 1,4 glycosidic linkage because of the numbering in the two glucose molecules. The 1,4 glycosidic linkage, and it will form a maltose. And for glucose and fructose, they get linked. So they'll also form a dehydration reaction in the synthesis of sucrose. That will be a 1,2 glycosidic linkage reaction that will give rise to the formation of sucrose. In other case, we have polysaccharides, which will be the polymers of sugars basically, and they'll have mostly storage and structural roles. And the structure and function will be determined by the sugar monomers and the positions of these glycosidic linkages. So for example, the most prominent one will be starch, which is a storage polysaccharide of plants, and it consists entirely of glucose monomers. And we know that plants obviously store surplus starch as granules within the chloroplast and other plastids, and the simplest form of starch is amylose. So we have both amylose and amylopectin. If you can see, this is the amylose, which is the simplest form of starch, and we have amylopectin as well, which is branched form of starch. Also, we have starch is a plant polysaccharide. We have glycogen, which is an animal polysaccharide. You can see the structure over here. And it's storage polysaccharide in animals. So humans and other vertebrates can store glycogen mainly in their liver and muscle cells. Okay. So another structural polysaccharide that is of importance will be cellulose, which is a major component of the tough walls of the plant cells. And like starch, cellulose is a polymer of glucose, but the glycosidic linkages will differ. Okay, because the difference is between the ring forms for glucose. If you can see over here, these are the alpha and the beta glucose structures. If you can see, and these are alpha and the beta glucose ring structures. And in case of starch, we see a 1,4 linkage of alpha glucose monomers. But in case of cellulose, we see a 1,4 linkage of beta glucose monomers. So if we basically the nomenclature, the key difference between these two lies between the orientation of the hydroxyl group, which is on the same side as in case of alpha glucose. If you see, so the main difference between alpha and beta glucose is that the hydroxyl group is on the same side in case of alpha glucose. If you see over here, okay, and this, but it's on the opposite side in case of beta glucose. The alpha glucose is more reactive to enzymes, and because of this, enzymes easily break alpha glucose. So alpha glucose is less stable as well. Okay. So the orientation of OH and H carbons on one, this basically determines if it's alpha glucose or beta glucose. Okay. So polymers with alpha glucose are helical, polymers with beta glucose are straight. And in straight structures, the H atoms on one strand can bond with the OH groups on the other strands. Correct? And the parallel cellulose molecules, yeah? Yeah. Sorry, I cannot hear a single thing. Yeah. Yeah. In alpha glucose. Okay, so I think the alpha glucose, wait a second. So in case of a cyclohexane ring, basically, as far as my knowledge goes, substituents in a cyclohexane ring will prefer to occupy the equatorial positions rather than the axial positions, so that the steric strain is basically minimized. From these structures we'll not be able to say, from the chair form structures we'll be able to say this better. So in beta glucose, all the substituents will probably occupy the sterically preferred equatorial position. Okay, and that is why beta glucose is most widely distributed sugar in nature. But in alpha glucose, all the substituents will be like equatorial apart from the hydroxyl group on the anomeric carbon C1, and will be on the axial. So that is why the most stable beta anomer actually predominates by 64 to 36%. In if we talk about an equilibrium of glucose, the beta glucose is more stable. I think this comes because of the So this structure has a symmetrical equatorial axis. So the substituents of the glucose ring which will extend outward or perpendicular to the equatorial axis will be the alpha glucose, and the substituents that extend along the equatorial axis will be the beta glucose. Okay, so the substituents extending along symmetrical axis is expected to have less steric hindrance, hence more stable conformation. So obviously that cannot be seen from these structures. Obviously chair form structures will be more important, and obviously another reason can be I guess in intramolecular hydrogen bonding between the H of hydroxyl group of anomeric carbon atom of beta with the near oxygen atom. Okay, so the OH group, the OH group here at position 1 of the alpha glucose, it will be occupying the axial position, while in this case it will be occupying the equatorial position in the respective chair conformations. I don't have chair conformations images here. So as a general rule, we know that the bulky group will tend to be more stable in the equatorial position, so to minimize steric hindrance. So I guess that is one of the reasons why beta glucose, beta conformation is more stable. Yeah. Thank you. Yeah. So polymers with alpha glucose are helical, and polymers with beta glucose are straight. And obviously the parallel cellulose molecules always held together in this way are grouped into microfibrils, which obviously form strong building materials in plants. Moving on. So you can see this is cellulose and this is a cell wall. So these are beta glucose monomer linked by I think 1 glycosidic linkages, 1 for glycosidic linkages in cellulose, and these basically this has been a schematic that is being shown here. So enzymes that can digest starch by hydrolyzing alpha linkages cannot hydrolyze the beta linkages in cellulose. So cellulose in human food can pass through the digestive tract as insoluble fiber. So there are some microbes that have enzymes to digest cellulose, and many herbivores from cows to termites have symbiotic relationships with these microbes. Okay, so we cannot really digest cellulose because our enzymes are not capable of hydrolyzing these beta linkages. We can talk about another structural polysaccharide, chitin, which is basically found in the exoskeleton of arthropods, such as cockroaches. The exoskeleton of cockroaches, arthropods will be of chitin. And chitin also apparently provides structural support for the cell walls of many fungi. Okay, and apparently chitin is also application-wise. They are used to make strong and flexible surgical thread that can decompose after the wound or incision heals. Okay, so coming from carbohydrates to lipids, lipids are obviously a diverse group of hydrophobic molecules. So they are one class of large biological molecules that do not form polymers. Okay, and they have little or no affinity for water. So why are they hydrophobic? Because they consist mostly of hydrocarbons which form non-polar covalent bonds. And the most biologically important lipids will be fats, the phospholipids which form the membranes, the bilayer, phospholipid bilayer which form the cellular membranes, and steroids. And fats can be constructed from two types of smaller molecules, either glycerol or fatty acids. So glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon, whereas fatty acid will consist of a carboxyl group attached to a long carbon skeleton. So if you see this, glycerol is this 3 carbons. So there are 3 carbons, and this is the glycerol, and this will be a fatty acid, which is obviously a huge hydrophobic molecule. And okay, so there can be ester linkages, so there can be dehydration reactions that will lead to the synthesis of fat. So if you see, there are ester linkages forming from the three basically linking the glycerol to the fatty acids, and this will lead to a fat molecule, and we can call this triacyl glycerol. Okay, there are three acyl chains, triacyl glycerol. Okay. Now fats will separate from water because water molecules form hydrogen bonds with each other, and they'll exclude the fat. And in fat, there are three fatty acids are joined to a glycerol, which we have already discussed, and all of them are ester linkages. This will form a fat molecule, triacyl glycerol. Okay, this is the structure of triacyl glycerol. Now these fatty acids can vary in length, obviously, because the number of carbons will differ, and obviously the number and the location of the double bonds will also differ. So the location and the number of double bonds will basically talk about the unsaturated portion of fatty acids. So saturated fatty acids will have the maximum number of hydrogen bonds possible and no double bonds, whereas unsaturation can be judged by the number of double bonds if it has one or more double bonds. Okay, so this is an example of a saturated fatty acid. If you can see the structural formula of a saturated fat molecule, okay, there are no double bonds here as such. But in case of unsaturated fatty acid, this is because there are double bonds. So there is a cis double bond that causes bending. So this is what unsaturation is all about, the number of double bonds. So fats made from saturated fats are called saturated fats, and they are mostly solid at room temperature like butter. And most animal fats are saturated. But fats made from unsaturated fatty acids are liquid at room temperature. So plant fats and fish fats are usually unsaturated. Okay, so the building blocks of unsaturated fats, these have bends or kinks, okay, because of the double bonds, and these don't allow the blocks to be tightly stacked, and hence they appear more fluid and liquid at room temperature as compared to the saturated fats. So due to the decreased saturation with hydrogen bonds, the structures are weaker and therefore typically oil like liquid in room temperature, and they are mostly found in vegetables as well as fish. Okay, so a diet rich in saturated fats may contribute to cardiovascular diseases through plaque deposits. So you will have arteries, right? So there might be plaque deposits on your arteries, which is not a good sign, obviously. So that might lead to various kind of cardiovascular diseases. So hydrogenation is the process of converting unsaturated fats to saturated fats by adding hydrogen. Okay, so if you want to convert something unsaturated to saturated, we'll have to remove the double bonds, and to remove the double bonds you have to add hydrogen. Correct. And hydrogenating vegetable oils also creates unsaturated fats with trans double bonds. So these trans fats apparently contribute more than saturated fats to cardiovascular diseases. Okay, certain unsaturated fats are not synthesized in the human body and they need to be supplied. So for example, omega-3 fatty acids, they are required for normal growth and are thought to provide even protection against cardiovascular diseases. These are found in various fishes, as far as I've heard. So these need to be supplied in the diet in order to provide protection against cardiovascular disease. The major function of fat is obviously energy storage, and for us, for humans and other mammals, we can store our fat in the adipose tissues. And adipose tissues, they can also they're a fat layer basically, and so they can cushion our vital organs from any kind of injury, and they can also insulate our body. Coming to phospholipid, which is one of the most important lipid categorizations, because they have two fatty acids and a phosphate group attached to a glycerol, and the fatty acid tails are hydrophobic, but the phosphate group and its attachments form a hydrophilic head. So if you can see, this is the hydrophilic head, and these are the tails which are hydrophobic. Okay, so basically these formed a bilayer on the cell surface membrane. The hydrophobic head will be basically inside, and the hydrophilic tails will be outside. Okay, so when they're added to water, they self-assemble into a bilayer with the hydrophobic tails pointing towards the interior. Obviously, because they are not water-loving, they are not hydrophilic. The structure of phospholipids will result in a bilayer arrangement in the cell membranes. So these are the major components of all cell membranes. So see, this is the hydrophilic head is projecting outwards because they are water-loving. The hydrophobic heads all come together to form a hydrophobic interior, and the hydrophilic head will face the water. Steroids are also lipids categorized by carbon skeleton, and they have 4 fused rings. Cholesterol, which is an important steroid, is also a very important component of animal cell membranes. Apparently, cholesterol helps in, cholesterol is found in lipid rafts. They help in better packing or more ordered cell membranes. So although cholesterol is essential in animals, high levels in the blood may contribute to cardiovascular diseases. Okay, this is the structure of cholesterol. Okay, coming to proteins, proteins account for more than 50% of the biomass of the cells, and you do not need to discuss how important proteins are, because proteins have diverse functions. Okay, so that is why people in the earlier days actually had this dilemma that proteins might be the genetic element, because they are so diverse. They give structural support, storage support, transport, cellular communication, movement, defense against foreign organisms, etc. Everything can be given by proteins. Okay, so we have enzymatic proteins. So proteins can act as enzymes. What are enzymes? Enzymes are biological catalysts. Basically, they will, in any kind of reaction, they will basically lower the activation energy barrier, and basically it will accelerate chemical reactions, such as digestive enzymes catalyze the hydrolysis of bonds in food molecules. We have defensive proteins for protection against diseases. Antibodies inactivate and help destroy the viruses and bacteria. So we have proteins that are in the immune system as well. We have definitely storage proteins like casein. The protein of milk is a major source of amino acids for baby mammals. Okay, storage of amino acids protein is obviously helpful as a storage material. And we have transport proteins like hemoglobin, the iron containing protein of vertebrate blood. It transports oxygen from the lungs to the other parts of the body. So other proteins transport molecules across the cell membrane. So proteins have diverse function. Okay, so we have hormonal proteins, receptor proteins. Receptor proteins basically respond to a cell or to a chemical stimuli. So receptors built into the membrane of a nerve cell can detect signaling molecules released by other nerve cells. If you can see, these are the receptor proteins, and they're just signaling molecules that are released, and the proteins can perceive that. We have hormones. Hormonal protein like insulin, which is a hormone secreted by the pancreas, causes other tissues to take up glucose and that helps in regulating the blood sugar concentration. We have contractile and motor proteins that help in movement. Proteins that are responsible for the undulations of cilia and flagella. Actin and myosin proteins are responsible for the contraction of muscles. We have structural proteins which help in giving support, like keratin. Okay, keratin, which is found in your hair. Keratin is the protein of hair, horns and feathers, and we have collagen and elastin proteins as well, which give a fibrous network in animal connective tissues. Okay, so enzymes are basically a type of protein that can act as a catalyst to speed up chemical reactions, and enzymes can perform their functions repeatedly, functioning as workhorses, and they can carry out most functions. So what are polypeptides? Polypeptides are unbranched polymers built from the same set of 20 amino acids. But protein is a biologically longer functional molecule that consists of one or more polypeptides. That is basically the difference in this nomenclature, difference between polypeptides and proteins. The monomers will be amino acids, which are basically organic molecules with one carboxyl and one amino group, and obviously a side chain. Okay, so the difference in the properties of the amino acids rise because of the differing side chains, which are the R groups. For example, in glycine, we have a hydrogen in place of the R group. That is the simplest amino acid. Lysine, arginine, histidine, these are positively charged or basic amino acids. Then we have aspartic acid, glutamic acid, these are acidic, negatively charged amino acids. So everything depends on the side chains or the R groups, which basically give each amino acids their properties. Okay, this is the structure of an amino acid. We have an amino group. This is the alpha carbon. We have a carboxyl group, and we have a side chain. So this is the structure of glycine, the simplest amino acid. So we know about the different amino acids. We have hydrophobic amino acids with non-polar side chains. We have polar side chains, so hydrophilic amino acids, and we have electrically charged side chains, like as I said, aspartic acid and glutamic acid are acidic or negatively charged, whereas lysine, arginine, and histidine are basic or positively charged. Okay. So amino acids can be linked. How do these monomers form polymers, basically proteins or polypeptides? So amino acids are linked by peptide bonds, and this peptide is a polymer of amino acids, and they can range in length from a few to more than 1,000 monomers. Okay, and each peptide, obviously, so there are various amino acids. At one end there will be a free carboxyl end, a free carboxyl functional group, and one end, at the other end there will be a free amino functional group. So the amino functional group end is called the N terminus, and the opposite end, which has a free carboxyl group, will be the C terminus. Okay, so these are two amino acids. So the peptide bond CONH, the peptide bond is formed like this by releasing H2O. Okay. So if you see, this will be the amino end, amino terminus, this will be the carboxyl end. If this is a polypeptide, basically 3 amino acids. So this has a functional, this is the backbone, these are the side chains. So this, at this end the amino group is free, so N terminus or amino terminus, and in this end the carboxyl end is free, so carboxyl end or C terminus. Okay. Our functional protein is a very complex structure. It will not consist of only one polypeptide, probably more than one polypeptide, and the most important thing is its folding. It will be twisted, folded and coiled into a very unique shape that will be very prominent and compatible with its function. Okay. So having a protein structure properly means the amino acids need to be proper, and the folding also needs to be proper. If you have a proper length of amino acid with exactly the proper sequence, even then the proteins might not function if the folding is not proper. So folding of proteins is very important for their compatible functions. Okay. So the sequence of amino acids can actually determine a protein's 3D structure, and a protein structure will obviously determine its function. Okay. So a little bit about the 4 levels of protein structure. The primary structure of a protein is its unique sequence of amino acids. The secondary structure is found in most proteins, consists of coils and folds in the polypeptide chain, and the tertiary structure is determined by interactions among the various side chains of the R groups. Whereas the quaternary structure is basically when the protein consists of multiple polypeptide chains, like multiple subunits. If a protein has multiple subunits that come together and basically interact by disulfide bonds or any kind of bonds and form a particular structure, particular folding, those will lead to the quaternary structures. So this is the primary structure, the sequence of amino acids. Like a line of amino acids, only the sequences of amino acids are taken into account. So basically like the order of the letters in a long word, and it's determined by what determines the primary structure of proteins? Obviously the genetic element. Okay, so the DNA content of your cell will determine the amino acid sequence. Okay, we know about the genetic code. Then comes secondary structure. The most important secondary structures are alpha helix and beta sheets. Obviously we have beta loops and turns and all of that, but these are the most prominent structures. This is secondary structure. It can either be alpha helix or beta sheet, which will again fold upon each other to form the tertiary structure, a 3D basically arrangement of the polypeptides, and then 2 or more polypeptides or subunits can come together to form the quaternary structure. This is the 4th level of protein structure. So the coils and folds of the secondary structure can result from hydrogen bonds between repeating constituents of the polypeptide backbone, and typical secondary structures are coil called alpha helix or folded called beta sheet. If you can see over, this is alpha helix. We have hydrogen bond between one amino acid and the fourth amino acid in the row. So that these are hydrogen bonds. In case of beta pleated sheet, these are beta strands. They are shown as flat arrow pointing towards the carboxyl end. Okay, and there are hydrogen bonds between the beta sheets. The tertiary structure is obviously determined by interactions between the R groups rather than the interactions between the backbone constituents. Okay, and basically it's a 3D. It's basically putting it in a 3D rearrangement. And these interactions between the R groups can include hydrogen bonds, hydrophobic interactions, van der Waals, ionic bonds, etc. And there are some strong covalent bonds also called the disulfide bridges, which basically reinforce the protein's structure. This is the tertiary structure. Then these are if you can see, these are hydrophobic interactions and van der Waals interactions. We can have hydrogen bonds as well. We can have disulfide linkages as well. We can have some kind of ionic bond as well. So the polypeptide chain folds upon itself and forms these kind of interactions and ultimately can give rise to the quaternary structure. Okay, so these can be, depending on the type of protein, these can be consisting of 2 subunits, 3 subunits, or more than 2-3 subunits. And basically in this case, for example, in transthyretin protein, there are 4 identical polypeptides that come together, fold upon each other and form the quaternary structure. Okay, these are some structures of proteins. This is a collagen structure, this is hemoglobin structure. So quaternary structure basically results when two or more polypeptide chains form one macromolecule, and the collagen is a fibrous protein consisting of 3 polypeptides coiled like a rope. Whereas the hemoglobin is a globular protein. If you can see over there, it's a globular protein consisting of 4 polypeptides, 2 alpha subunits, this is alpha, and 2 beta subunits. Okay. So a little bit into sickle cell disease. A little change, a little mutation in the primary amino acid structure can lead to such a disastrous disease. Okay, because it will, a little change will change its structure, its function, and hence lead to the disease. So this sickle cell disease is basically an inherited blood disorder, and it results from only one single amino acid substitution in the protein hemoglobin. Okay, so in the normal hemoglobin, this is the primary structure. If you can see over here, the 6th position we have GLU. Okay, but in case of sickle cell and hemoglobin, we have valine. Okay, and because of this, the secondary structure will not form properly because there will be an exposed hydrophobic region. The quaternary structure will not form properly. This is the normal hemoglobin, this is sickle cell hemoglobin. And because of that, the function is affected, because in case of normal hemoglobin, the molecules do not associate with one another, each carries oxygen. However, in case of sickle cell hemoglobin, the molecules will crystallize into a fiber, capacity to carry oxygen is reduced. So in RBCs, normally RBCs are like this, but sickle cell hemoglobin containing, like a sickle cell disease person's hemoglobin will be like this. It's sickle shape. It is how they got the name sickle cell anemia, sickle cell hemoglobin. So what determines the protein structure? In addition to the primary structure, some physical and chemical conditions can also affect the structure. Like if you have a protein that is properly folded, but we change the pH, salt concentration, temperature, anything around it, it will obviously cause the protein to denature, to unravel. Okay? And a denatured protein cannot act like how it was functioning before. So denatured protein is biologically inactive. This is a normal protein. If we change the conditions, its surrounding conditions to different pH or different, if we add different, if we add a lot of, if we increase the temperature, basically it will denature our protein. Now it is hard to predict the protein structure from its primary structure. Most proteins probably go through several stages on their way to a stable structure. So there are something called chaperonins, which are protein molecules that assist the proper folding of other proteins. Okay. Chaperones are found in the cells, and they basically help your proteins to fold properly. So there are diseases such as Alzheimer's, Parkinson's, mad cow disease, and all of these are associated with misfolded proteins. So you can see even misfolded proteins can cause mess in your cellular system, because misfolded proteins will not be carrying out their functions properly, and that will lead to very reduced functional property of the enzymes or the proteins, and that will ultimately lead to these kind of diseases. Okay, so this is a example of a chaperonin. What happens is that the polypeptide which needs to be folded, it's an unfolded polypeptide, basically enters the cylinder like chaperonin structure from one end, and the cap also attaches, causing the cylinder to change shape in such a way that it creates a hydrophilic environment for the folding of the polypeptide. Ultimately, the correctly folded protein is thrown out, the cap comes off, and the properly folded protein is released. So this is the function of a chaperone. Okay, so this is the function of a chaperone. Now, how do we know the protein structure, the native protein structure? So scientists have used various techniques such as X-ray crystallography, NMR spectroscopy. People are doing cryo-EM, which is another revolutionary technique in today's time. And there are obviously bioinformatics studies, in silico studies, where people predict protein structures from the amino acid sequences. So we have the amino acid sequence, we know the hydrophobicity and hydrophilicity of all the amino acids. From there, the bioinformatics software basically they try to predict the protein structure and protein folding. Okay. X-ray crystallography is very popular. So there's an X-ray source, X-ray beam. We have to create crystals of the protein, and the diffracted X-rays will be taken by the digital detector, and from the X-ray diffraction pattern one can try to create the protein structure. Okay, we have talked about nucleic acids previously. We know that nucleic acids store, transmit, and help express hereditary information. and the amino acid sequence of a polypeptide is also encoded by the gene. So the gene, so anything, even a protein's primary structure and amino acid sequence is dictated by the gene. So if our gene is working, if our gene is not mutated, hopefully the protein will also not be mutated. Okay, and genes are made up of DNA and basically monomers called nucleotides, which come together and form stacks, basically called the polymer on DNA. So we have talked about the two different types of nucleic acids already: deoxyribonucleic acid and ribonucleic acid. And DNA can provide direction for its own replication via semi-conservative replication. And from DNA we get mRNA. From mRNA we can get protein. That is a central dogma. There are some instances where from RNA we can get DNA, cDNA, and that is basically carried out by some enzymes known as reverse transcriptase. Right? The protein synthesis occurs in the ribosome. So the DNA is inside the nucleus, but it transforms into, it basically converts into mRNA or messenger RNA, and then goes into the cytoplasm and basically attaches to ribosomes, from which protein synthesis can occur. Okay? DNA synthesis of mRNA, it goes movement into the cytoplasm, and then from ribosome we have the polypeptide, synthesis of protein. So nucleic acids obviously polymers and form of polymers called polynucleotides, and each polynucleotide will be made up of monomer. It will be made up of monomer nucleotides, which we have already talked about. We have already seen the structures as well, correct? The nitrogenous base, the pentose sugar, and one or more phosphate groups. Okay, so the portion of nucleotide, if the phosphate group is not being considered, it is called nucleoside. But nitrogenous base, pentose sugar, and phosphate group, three of them together are called nucleotide. Okay, without the phosphate group, it is called nucleoside. You will see over here. So this is a proper nucleotide. This is the sugar, pentose sugar, the nitrogenous base, and the phosphate group, the pyrimidids and the purines. Sugars different because in RNA we have the ribose sugar, and in DNA we have the deoxy ribose sugar. And we also know that DNA is obviously more stable than RNA. Okay, because due to deoxyribose sugar, it contains one less oxygen containing hydroxyl. Okay, and since DNA is more stable component, it is useful that we keep a stable component. We give a stable component the role of keeping genetic information safe. Okay, RNA containing a ribose sugar is more reactive than DNA, and it's not stable in alkaline conditions at all. So that makes DNA the stable element, and DNA is basically what has evolution has chosen to be the genetic element of our system. Okay, so we have already talked about the pyrimidins and the purines, and DNA, the sugar is deoxyribose, RNA it is ribose. Okay, they are linked together to form the polynucleotide chains. Now the adjacent nucleotides are joined by covalent bonds that will form the OH between the OH group of the 3' carbon of one nucleotide and the phosphate group of the 5' carbon on the next. And these basically links will create the backbone of the sugar phosphate units, and the nitrogenous bases will be hanging out, correct? We discussed in one of the previous slides, the backbone will be because of the sugar phosphate, the backbone will be because of the sugar phosphate linkages, and these will be what are hanging out. The sequence of bases along a DNA or mRNA is unique for each gene, because each gene with this nucleotide sequence is basically dictating how the DNA will look like, and then how mRNA will look like, and finally how the protein will look like. Okay, RNA molecules on the other hand, they exist usually as single polypeptide chains, not really a double helix model like DNA. DNA exists in a double helix model, forming spiraling around an imaginary axis. Okay, and in the DNA double helix, as I said before, the two backbones, the two strands of the DNA double helix are anti-parallel to each other. So they run in opposite 5' to 3' directions. Okay, and the one DNA molecule will obviously include many genes. We have talked about this also, that adenine always pairs with thymine with 2 hydrogen bonds, and guanine with cytosine with 3 hydrogen bonds. This is known as complementary base pairing. So the complementary pairing can also occur between the two RNA molecules or between parts of the same molecule. In RNA, the thymine is not there. In RNA, it is replaced by uracil. So in RNA, the adenine and the uracil will pair because there is no thymine in RNA. Okay, this is DNA, and you can see the sugar phosphate backbones, the hydrogen bonds, the base pair joined by hydrogen bonding. Okay, so the linear sequences of nucleotides in the DNA molecules are passed from parents to offspring, and if we talk about two closely related species, they are more similar in DNA content than more distantly related species. So molecular biology and studies can be actually used, phylogenetic studies and all of these can be used to assess evolutionary relationship as well. Okay. So you have talked about all the different carbohydrates, lipids, proteins and nucleic acids and their basic roles, and we have talked about carbohydrates being the fuel and the building materials, the different types of carbohydrates. We have talked about lipids, phospholipids, steroids. We have talked about triacylglycerols. We have talked about proteins giving a diversity of functions like structural, enzymatic function, and nucleic acids. The basic role will be stability, which gives it the role of transferring genetic information from parent cell to offsprings. Yes. Okay. So these are not really needed. So we have talked about carbohydrates as well, and the glycosidic linkages between different carbohydrates. Lipids we have talked about, they don't interact, they have very less affinity for water, which is why they are hydrophobic. They are polymers made up of fatty acid monomers. We have talked about the lipid layers. The hydrophilic head group will be pointing outside towards the water, and the hydrophobic tails will be inside. Okay. So you talked about nucleic acids. So the question that we started with was which element transfers genetic material from one cell to another, one cell to their offspring. So that will be nucleic acids. Okay. Moving on to the next question for today. What is the energy carried by a photon of wavelength 700 nanometer? And we have been given the values, speed of light as well. So will anybody like to try? So we have been given the values of Planck's constant and the speed of light as well. Second option. Anybody else? Yeah, so if we put the basic formula that we have, we can get the answer. So we know that light acts like a wave. It can be like thought of a propagating electromagnetic wave, and the wave will travel at a speed 10 to the power 8 meter per second through vacuum, and the electric and the magnetic fields are basically felt to oscillate at right angles to each other as the wave as the wave passes. Okay, and the distance between the crests or the troughs can be called the wavelength. So if the wave travels at speed c, the crest will pass through with the frequency f. So this is the relation between wavelength and frequency and the speed of light. Okay, so light can also act as a particle. That is a dual property of light. It can also act as a particle, the photon. So when detectors see light, it arrives in discrete packages which we call photons. So you can think of each photon as having a wavelength, and the energy of a photon depends on its wavelength or frequency. Okay, c by lambda. And as a whole, the photons have statistical behavior of waves of that wavelength. So they experience interference like waves. Okay. So light is an electromagnetic radiation, and all wavelength or energies of light are the same basic stuff. Together they constitute this entire electromagnetic spectrum. Okay, the visible light is only this much tiny portion of this. Our eyes can detect only the visible light, but the rest of the spectrum cannot be detected by our eyes. But now we have detectors that can. So we give different names, different colors. We use different units for the different wavelengths, whatever is convenient. So only this part of the spectrum is what we can basically judge with our eyes. But the rest of the spectrum, which cannot be, we have today scientists have built detectors for the same, but we cannot detect that with our eyes. So this is the electromagnetic spectrum. Okay, so apparently astronomical objects are capable of producing different parts of the spectrum depending on how energetic the processes that are going on and how hard the objective is. Okay, so we know about the different laws, and we will basically move on to the equation that tells us the wavelength, basically equation between the energy and the wavelength. So we know that the energy carried by a photon is given by this quantum relation, energy equal to h nu, and interchangeably energy equal to hc by lambda. C is the speed of light, lambda is the wavelength. So if we put all the formula over here, we were given the Planck's constant, we were giving the speed of light, and the wavelength was 700 nanometer. Okay, so you have to put that in meters, so 10 to the power minus 9. So if you do this calculation, the value will be around this. Okay, so the answer will be the second joule, 2.8 into 10 to the power minus 19 joule will be the answer. Okay, moving on to the next question for today. So what happens to animal cells when they are placed in pure water? Anybody? Take in water and swell. Okay. Anybody else? Why will they take in water and swell? What is the logic? So the solutes will be more in the cell than in the water. So the water will move from the water basically will move from the outward pure water to inwards inside the cell, and the cells will swell. They'll take in water and swell. Yeah. So basic definition of diffusion and like movement of movement of molecules, any molecule from a region of higher concentration to a region of lower concentration, and if that needs use of energy, especially when it is going against the concentration gradient, we can call that active transport. Okay, so obviously in cells there are movement through different membranes, and all the membranes in a cell can act in the cell in the same way. So membranes can be classified into permeable, basically that is very that's not selective at all, that can let everything in and out. So whatever is on both sides of the membrane can be taken in and can be thrown out. There is semi-permeable, which basically lets some things in and out, and impermeable, which does not let anything come in or go out. Okay, so example of diffusion in everyday life is, if we spray a bottle of perfume at one position, the person sitting a little away can also smell the perfume because of diffusion. Correct. Osmosis is the movement of water across a semi-permeable membrane from a region of high water concentration to a region of low water concentration. Okay, so that is basically what will happen. So the cells will have a lot of solutes, a lot of concentration of solutes inside. So there will be osmosis because the water will move from the pure water thing to the inside the cells. So from a region of higher concentration to a region of lower water concentration. Okay, and osmosis that does not really require energy, so it is passive. So exactly this is what will happen. So if this is a selectively permeable membrane, and this portion has high sugar concentration. Sugar is shown by the green circles, and low water concentration. However, the other portion has high water concentration and there is almost no or very low sugar concentration. Okay, so osmosis, how will it happen? So the water will move from across the permeable membrane from its higher concentration to its lower concentration. Okay, so if pure water is separated from salt water, what will happen? The water molecules will move randomly on both the directions, and the salt molecules cannot move out. So more water molecules, so the membrane is semi-permeable. So the salt molecules are not allowed to go here and there. The salt molecules cannot really move out. So more water will move into the salt solution. So what you will have is water moving from where there is higher concentration of water to where there is low concentration of water. So this will basically what we call osmosis. Correct. So we know that animal cells are enclosed by membrane, and if an animal cell is surrounded by a solution that is the same concentration as the cytoplasm in the cell, water will move in and out at the same rate and the cell will remain intact. Okay, so if you want to keep an animal cell into a solution and still ensure that it is not shrinking or it is not swelling, then we have to ensure that the solution we are putting it into has the same osmotic pressure, the same concentration as the cytoplasm in the cell. So in that case, water will both move in and out. Basically, there will be an equilibrium and the cell will remain intact. Okay. So we can imagine, so there are so many animals, marine organisms that live in the sea. Okay, and sea will have, like, it's basically salt water. So how do they survive? Ideally, if the organisms have lesser solute concentration as compared to the salt water, all the water should move out and they should shrink. But there are animals which have similar cytoplasm with similar concentration to seawater. Okay, and in our case, it is the function of our kidneys to make sure that the fluid in our bodies that surrounds our cells has a stable concentration. So if an animal cell is in a solution that is less concentrated than its cytoplasm, that is more pure water outside than inside the cell, then the water from outside will move into the cell and it will swell and swell, and at some point it will cause the cell to burst and die. Correct? So, for example, and if animal cells are in a solution that is more concentrated, if we do not keep it in pure water as was the question, and if we keep it in a solution that is far more concentrated than the cytoplasm, so that is there's more water in the cytoplasm than in the solution outside, then the water will leave the animal cell and the cells will shrink, shrivel and die, because there is more concentration of solutes outside. So all the water from the cells will go out into that solution. Okay, so basically this is what happen when there's H2O coming inside, and inside ultimately it will burst. This will be the hypotonic shock. Okay, because water is coming inside the cells and ultimately the cells are swelling and swelling, and at some point it bursts. If the concentration on both the sides are maintained intact, so there will be equal moving in and out of water, so in equilibrium will be maintained, so that will be an isotonic situation. Okay, but if there is a scenario where the outer medium is very concentrated and more concentrated than the cells, the water from the cells will move out. Okay, and the cells will start shrinking and it will ultimately shrivel and it will go through this hypertonic shock. Okay, now plant cells have a membrane and is also surrounded by a cell wall. So in animal cells, the difference is that we don't have a cell wall. In animal cells, is only a cell membrane, but plant cells have a cell wall. So cell walls are fully permeable, and they allow all substances in and out of the cell. So if a plant cell is surrounded by less concentrated solution, then water from outside will move into cytoplasm and the vacuole of the plant cells. So this is basically how roots absorb water by osmosis, right? And the water outside, if is less concentrated than the water in the cytoplasm, again, when the outside water enters the plant cell, the vacuole will become bigger and the cytoplasm will swell. Correct? And this will cause the membrane to be pushed towards the cell wall, which will cause the turgor pressure. And when cells are fully swelled like this, the membranes will be pushing against the cell wall and they will be described as turgid. If you see over here, this is the isotonic solution. Isotonic, so intact, both sides is intact. But in this case, the water will, if it is a less concentrated solution outside, water will come in and ultimately the cell membrane will push towards the cell wall and it will become turgid. On the other hand, if the opposite happens, the outer surface is much more concentrated, then water will go out of the vacuoles of the plant cell and the plant cells will basically shrink and plasmolize, will be plasmolized. Okay, so this turgor pressure is what gives plants their strength. If plants did not have this, they probably would have wilted. They would have wilted, not probably, they would have wilted. And plants that don't have wood, such as lettuce and houseplants, may basically rely on turgor pressure for strength. Okay, so if plant cells are surrounded by a more concentrated solution, like salt water, the water inside the cell will move out to the more concentrated solution outside and it will basically get plasmolized. Right? The water from the vacuoles and the plant cells will go outside and the cells will become plasmolized. So when this happens, the cell wall stays intact but the membrane shrivels up away from it, and this is called plasmolysis. So cells in this condition are called plasmolized cells. So we can basically look into it by placing a layer of red onion cells in salt water. We can see how the cells start shrinking and shriveling because the water content is going outside. Okay. Now, bacteria and fungi are the most common causes of food spoilage, and both these cells are enclosed by a membrane. So if food is placed in a sugary or salty solution, then any bacteria or fungi present will lose the water in their cells to the more concentrated solution outside. Correct? So when this happens, the cells will shrivel and die and the food will not become contaminated with loads of bacteria and fungi. Okay? So that's why fish, meat and all of these may be stored in a salty solution. Okay, and jams, marmalades, and tinned fruits they are stored in sugary solution. So everything has an application. So you have already talked about water potential and osmosis. Okay, for water potential, basically it's denoted by psi, this Greek symbol, and it's basically tendency of solution to take up water, like the tendency of water to diffuse from one area to another. Okay, and basically it will have two components: the pressure, obviously, and the physical forces, which can be negative or positive. If positive, increased pressure, and if negative, it is decreased pressure. Okay, and obviously the solute concentration, the osmotic potential. So at which compartments the solute concentration is more, which is always mostly negative. So water potential is the sum of solute potential and the pressure potential. It can be expressed as solute potential plus pressure potential. Okay, so pure water will have psi equal to 0. So adding solute will lower the potential because there are less free water molecules. Okay, and water will move from a higher water potential to a lower water potential. So a less concentrated to a more concentrated compartment. Okay. So water movement depends only on solute concentrations. Hypertonic solution, water moves out and the cell shrinks. Hypotonic, water comes in, swells, and ultimately bursts. Okay. So we have already discussed about this. For plant cells also, we have also talked about how they can exert pressure and how turgor pressure is an important way through which plants get their strength. Okay. So plants, it's important for plants because in some plants where there is no wood, like lettuce and house plants, this is how the plants maintain their strength. Okay, so you can actually carry this out in lab. In a beaker, we can put water and then sucrose, and then we can figure out what happens. We can add pressure from one side, unlike how we were studying reverse osmosis as well. If you remember, in one of the previous questions, we were talking about reverse osmosis for removing hardness of water. If you can think like that, you can put pressure and we can see how this osmosis things happen and what happens to the water potential that can be done in lab. Okay, so moving on to the next question for today. Which of the following is not a membrane bound cell organelle? Anybody else? Okay. Anybody else? Okay, so okay. So cell organelles can be bound by a single or double membrane. Nucleus, plastids in plant cells, mitochondria in both plants and animal cells are bounded by double membrane. Okay, whereas lysosomes, glyoxysomes, all of these are bounded by single membrane. So ribosomes are basically like not bounded by any membrane. They are not membrane bound and they can also float freely in cytoplasm. They can also be linked to the endoplasmic reticulum, which is how the rough endoplasmic reticulum gets its name. So the ribosomes can attach to the cytosolic side of the ER membrane, endoplasmic reticular membrane, and are engaged in the synthesis of proteins that are being currently translocated into the ER. There can be free ribosomes as well, which will be unattached to any membrane, and they can also synthesize all other proteins encoded by the nuclear genome. Okay, so these ribosome organelles, they do not require a membrane to carry their functions. So again, if we talk about the different cell organelles, we know that the interior contents of the cell will be the cytoplasm. Okay, bacterial cells are relatively simple cells because they lack a nuclear membrane. They have freely floating DNA, which is freely floating genetic element, which is known as a nucleoid, like nucleus, but they don't have proper nucleus, membrane bound nucleus, and obviously they lack various organelles. And the bacteria, these are all prokaryotes. For eukaryotes, we obviously have more complex cells. You have a lot of organelles, and the nucleus is properly the nucleus has two bilayers. Basically, it's a double membrane bound organelle. It has two bilayers, and so if you can see this is the nucleus, and we have a nucleolus, the nuclear envelope, and nucleoplasm, and all of that. So a eukaryotic cell is much more complex than a prokaryotic cell. Okay, so why are cells small basically? So as cell size increases, the volume increases much faster than the surface area. So cells obtain nutrients, gain information and get rid of waste through their plasma membrane only. So as the cell size increases, a cell's ability to exchange with its environment becomes limited by the amount of membrane area that is available for exchange. So that is why most cell size are around compatible, so that the surface area is perfect for the exchange of nutrients with the environment. Okay, so cell theory, we have already talked about this. It's a fundamental unit of life. Obviously, all biochemical processes are carried out by cells, and groups of cells can be organized, and they will form tissues if they function as multicellular structures in form, and we carry out sub processes of the multicellular organism. So prokaryotic or bacterial cell is very small with a plasma membrane surrounded by a rigid cell wall. In many, the cell wall is made up of a carbohydrate cross-linked with polypeptides. Okay, and cell wall may be covered by a capsule also made up of polysaccharides. So inside there are few or no membrane enclosed spaces, so no compartmentalization as such. So no nucleus. DNA is in a freely floating way called the nucleoid, and DNA is circular and naked because they generally don't have any protein associated with it. Okay, and basically membrane enclosed spaces allow the cell functions to be compartmentalized and isolated from other functions, but prokaryotes are very simple structures. They lack any kind of membrane enclosed spaces. So some prokaryotes can perform photosynthesis, and the biochemical machinery for photosynthesis, like trapping light energy, can be done within a highly folded plasma membrane. Okay. Now, eukaryotic cell structure, it's larger with a typical plasma membrane, and some have cell wall. The plant cells have cell wall, animal cells don't have cell wall, but there's a lot of compartmentalization inside because there are a lot of interior spaces enclosed by membranes such as nucleus, then the endoplasmic reticulum, Golgi apparatus, then we have the mitochondria, then we have chloroplast for plant cells, and we have lysosomes, which are the suicidal bags of the cells because they have a lot of hydrolytic enzymes. Then we have the vacuoles and vesicles, and obviously there is a cytoskeleton as well, which basically consists of microtubules, microfilaments, and intermediate filaments. Basically gives structure to the entire cell, structure and shape. Okay. So eukaryotic cells will have well-organized membrane-bound organelles and nucleus, and presence of the membranes will give a clear compartmentalization of cytoplasm. And they obviously will have complex locomotory and cytoskeletal structures. The cytoskeleton doesn't only give a structural shape; they also help in complex locomotory functions. And the genetic material is organized inside the nucleus. So the DNA is not freely floating or naked like in case of prokaryotes. They are linked to basic proteins such as histones, and they are condensed into chromosomes. Okay, so this is the basic structure of a eukaryotic cell. So if you can see, there are ribosomes in the rough endoplasmic reticulum, but there are ribosomes freely floating as well. Okay, so they are not really membrane bound. This is a plant cell. This is an animal cell. Okay. So firstly, we have talked about the lipid cell membranes. They are formed of a lipid bilayer, phospholipid bilayer. So the non-polar or the hydrophobic tail is protected from the aqueous environment, and the hydrophobic heads project out because they are water loving. And the ratio of protein and lipid obviously varies in different cells. So the lipid bilayer has it can have a peripheral protein like this. It can have a channel protein, a carrier protein, and there are different types of proteins that can be found here. So the integral proteins are partially or totally buried in the membrane, and there are peripheral proteins which lie on the surface of the membrane. Okay, so the Singer-Nicholson suggested the fluid mosaic model in 1972, and according to this, they said that the quasi-fluid nature of the lipid enables lateral movement of the proteins within the overall bilayer. Okay, so this ability to move within the membrane is measured as its fluidity. So these are actually done in experiments in labs. People try to treat cells with different drugs and check if the membrane fluidity is changing or not. So how do they do that? One of the experiments that can be done is that if some of the lipids are linked to fluorophores, and if one part of the fluorophore is bleached, and then at that portion they will try to see if there is any recovery of the fluorescence. So if there are other this part will be bleached. For example, if there are other fluorophore containing lipid molecules that are being laterally diffused, that are laterally diffusing into that region, then we can see a recovery in that area. So this is called FRAP, or fluorescence recovery after photo bleaching. So these are not only theoretical facts, people are actually doing such experiments in labs. So according to this model, the cruciform nature of lipid enables lateral movement of proteins within the overall bilayer. Then obviously there are cell membrane also allows transport of molecules. The cell membrane is selectively permeable to some molecules present on either side of it, and due to the fluid nature of the plasma membrane can help in cell growth, formation of intercellular junctions, endocytosis, etc. So there can be passive diffusion without the need of any energy, okay, and there can be facilitated diffusion, like requires the presence and the action of a channel protein. There can be active transport as well, where the molecule to be transported is transported from its, generally what happens is molecules go from their higher concentration to their low concentration. But if you want the opposite to happen, molecules can also be transported from their lower concentration to their higher concentration using energy. Okay, so ATP is utilized to ADP and inorganic phosphate. So this type of transport is known as active transport. Okay, so we have already talked about simple diffusion, movement of neutral solutes across the membrane, and osmosis, movement of water by diffusion across the membrane, across a semi-permeable membrane. But polar molecules cannot really pass through the non-polar lipid bilayer, so they require a membrane carrier protein. So that is why facilitated diffusion. So the transport is being facilitated by this channel protein. Okay. The movement of molecules across the membrane against the concentration gradient will obviously need energy. So that energy is being provided by ATP. So that is active transport. An example can be the sodium-potassium pump. Okay. I have talked about cell wall. It's found in plants and fungi. Okay, and cell wall of algae is found made up of cellulose, galactans, mannans, and minerals like calcium carbonate. In other plants, it consists of cellulose, hemicellulose, pectins, and different proteins as well. So the important function is, if you can see, it gives a shape and structure to the cell. Obviously, it protects the cell from mechanical injury and any kind of damage or infection. It can also help in cell-to-cell interaction, and obviously it acts as a huge barrier for any kind of macromolecules transport. Now coming to the endomembrane system. So endomembrane system is a group of membranous organelles having coordinated functions like endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. So all of these come under the endomembrane system. Okay, this is the nucleus, the rough endoplasmic reticulum, rough because the ribosomes are attached to this side of the ER. Then we have the Golgi apparatus, we have the smooth endoplasmic reticulum, which do not have any, you can see, which do not have ribosomes attached to it. We have the lysosomes and the vacuoles. So the endoplasmic reticulum is basically a network of like tiny tubular structures you can see in the image. They're scattered in the cytoplasm, and basically it divides the intercellular space into two compartments, the luminal inside the ER and the extraluminal, that is the cytoplasm. So they can be of two types, RER or the rough endoplasmic reticulum, and SER or the smooth endoplasmic reticulum. So these are required for lipid synthesis. RER is most commonly used in protein synthesis because ribosomes are there. Okay, so they have the RER bear ribosomes. They are frequently involved in protein synthesis and secretion, and they are continuous with the outer membrane of the nucleus. Okay, whereas in smooth endoplasmic reticulum, the ribosomes are absent, if you can see here, and here lipid synthesis occurs. In animal cells, lipid-like steroidal hormones are also synthesized in the SER. Okay, so it's basically a web-like series of membranes within the cytoplasm in the form of flattened sheets, sacs, and tubes. And they basically help creating compartmentalization, like membrane enclosed spaces. It spreads throughout the cytoplasm and has connections with the outer membrane of the nucleus. Its functions are circulation, transport, storage of proteins and minerals, synthesis of lipids, and obviously since it's folded so much, there is a large surface area for any kind of enzyme action. We have already talked about rough ER and smooth ER. Then you have the Golgi apparatus. Okay, so it was first observed by Camillo Golgi, hence the name. And basically the Golgi apparatus is a concentrically, you can see this is a concentrically arranged structure. Okay, so there is one cis phase, which is the forming phase, and a trans phase, which is a concave maturing phase. So they are totally different, but they are interconnected, if you can see. Okay, So basically the function is basically it secretes materials to the intracellular targets or outside the material. So anything that needs to be packaged as vesicles, they fuse, as vesicles, they from the ER they fuse with the cis phase. Okay, so from the ER they get out as vesicles, they fuse over here and move through all of this to the trans phase. Okay, so if you can see, this is an incoming transport vesicle. So it will come in through this and ultimately this will be a newly formed vesicle or a secretory vesicle. Okay, so the proteins that are formed by the ribosomes on the rough ER are modified in all these cisternae of the Golgi apparatus before they are released from the trans phase. The proteins can be made glycoproteins, lipids can be made glycolipids, so all of these happens. Then we move on to the lysosomes, which are basically also membrane-bound vesicular structures formed by the process of packaging in Golgi apparatus. And membrane is actually very important because the condition inside the lysosome is very acidic, as well as there are a lot of enzymes, hydrolytic enzymes. It basically contains almost all types of hydrolytic enzymes, and they are active at acidic pH. They digest carbohydrates, proteins, lipids, and nucleic acids. So anything that the cell wants to degrade, digest, they'll fuse that with the lysosome, and the lysosome contents will take care of that. So that is why lysosome is called the suicidal bag of the cell. Then we have the vacuoles. Even vacuoles are membrane-bound space found in cytoplasm, and it contains water, sap, excretory product, and other materials not really useful for the cell, and it is bound by a single membrane called tonoplast. So in plant cells, the vacuoles can occupy even 90% of the volume of the cell. You can see this is a plant cell, it can occupy even 90% of the cell. Okay, so in plants, this tonoplast facilitates the transport of ions and other materials against the concentration gradients into the vacuole, and hence their concentration is higher in vacuole than in the cytoplasm. Okay. In amoeba, however, the contractile vacuole has another function. It helps in excretion. And in many cells such as protists, there are food vacuoles that are formed by engulfing food particles. So these are the functions of vacuoles. Nucleus, obviously we have discussed. A cell has only one nucleus, and some mature cells like RBCs can even, mammalian RBCs can even lack a nucleus. Basically it is the control point of the cell, because your genetic material which dictates everything is inside the nucleus. It contains a nucleolus, which is a darkened region where the ribosomal RNA is synthesized, and it obviously contains the chromosomes, which is basically DNA. Each cell might have 3 meters to 4 meters of DNA, right? We have read somewhere. So imagine how the DNA is wrapped around the proteins, and it's condensed and folded so much so as to fit such a huge length of DNA in such a small space. The nucleus is surrounded by the nuclear envelope, which is a double membrane. Okay, double membrane, double bilayer, and the nuclear membrane has nuclear pores that control the entry and the exit of the materials. So interface is before the starting of the cell cycle. There is a resting phase, but people call it the resting phase, but there's a lot of stuff that is happening in the interface. So in the interface, nucleus will have the nuclear envelope, the nuclear matrix or the nucleoplasm, the nucleolus, and the chromatin. Okay, the chromatin basically, which ultimately gives rise to the chromosomes. The nuclear envelope is a double layered membrane. Okay, and there's a space in between called the perinuclear space, and it is basically a barrier between the nucleus and the cytoplasm. And this outer membrane, if you can see, it is continuous with the ER and also bears ribosome on it. And the nuclear membrane has pores, okay, formed by the fusion of the two membranes. And these are the passages for the movement of RNA. So we know that DNA will convert to mRNA, and mRNA will go out through these pores. Okay. Now the nucleolus is one or more non-membranous spherical bodies. It is continuous with the nucleoplasm, and basically in this part the ribosomal RNA synthesis happens. Chromatin is a network of nucleoprotein fibers. Okay, it contains the genetic element DNA and the basic proteins, and there are some non-histone proteins also, and RNA. So during cell division, the chromatins will condense and fold upon each other to form chromosomes, the condensed form of chromosomes, like how we see chromosomes structures like that. Okay, so the chromosomes will contain both DNA and protein, as can be seen under the microscope during the metaphase of cell division when it is the most condensed, and other times the DNA will be thread-like. Okay, and the most common proteins are the histone proteins or the basic proteins basically. So this is, so you have seen the chromatin structure, and from that it gets condensed to a structure like this chromosome. Okay, and every human cell has about like 2-meter long DNA, 2 to 3-meter long DNA distributed amongst its 23 pairs of chromosomes. Okay, so we have talked about the nucleus. Now ribosomes. Ribosomes are very important because they are a protein synthetic factory of the cell. They are of two subunits, the large subunit and the small subunit, and each will be made up of protein and ribosomal RNA, and the subunits will associate together. They will come together during the formation of proteins. So when the mRNA will go from between this, and tRNAs will also be involved, so the protein synthesis will occur. And basically the ribosomes, which are freely floating in cytoplasm and also attached to the ER, the rough ER, they help in protein synthesis. Okay. So the ribosomes are non-membranous granular structures. They do not have a membrane, and they are composed of RNA and proteins. Okay, and eukaryotic ribosome has two subunits, the 60S and the 40S. They together form the 80S. Then we talk about the Golgi apparatus, which is another member of the endomembrane system. It is a collection of membranes associated with the ER. We have already talked about this trans phase and the cis phase. The vesicles will come from the ER to the cis phase and then ultimately get released from the trans phase. So the transport vesicles will fuse with the Golgi and dump their contents into the Golgi, and the Golgi will package the proteins in the vesicles so that they may be excreted from the cells. Okay. And the Golgi apparatus can also form lysosomes. We have talked about lysosomes also, which are called the suicidal bags. And particles can be taken if there is a foreign pathogen, bacteria or anything that is coming. There will be phagocytosis by the cells, and the vesicle will fuse to the lysosomes, and the components of lysosome will basically digest or degrade that material. Okay. Then we have some other bodies like microbodies, peroxisomes and glyoxysomes, which are basically vesicles that form through growth and division in cytoplasm. And glyoxysomes can contain basically enzymes that convert fats into carbohydrates, whereas peroxisomes are basically used for removing any kind of reactive compounds from the cytoplasm. So they create hydrogen peroxide as a byproduct, and they can degrade it with the enzyme catalase. Okay. So coming to the mitochondria, which are obviously the part of the cell where energy is produced. So they have it's a double membrane structure again, with the outer membrane and the inner membrane, and it divides the lumen into two aqueous compartments. So this is the outer compartment, this is a matrix, this is the intermembrane space, and the inner membrane, if you see, there are so many foldings here which ultimately get the name cristae, and there are so many foldings in the matrix because they can increase the surface area. Okay, so the two membranes have their own specific enzymes associated with the mitochondrial function. So the matrix possesses the cellular DNA. So mitochondria has a DNA on its own, ribosomes, and even components of protein synthesis. So mitochondria are the sites of aerobic respiration. They can produce ATP, so they are called the powerhouse of the cell. Okay. Here chloroplasts, which are the sites of photosynthesis in nearly all plants and protists, and this is the structure of the chloroplast. This is outer membrane, inner membrane, and within the stromal matrix there is a series of stacked flattened membrane structures which are called thylakoids, and when these thylakoids stack upon each other, they are called grana. Okay, so the light energy trapping molecules of photosynthesis are basically found in the membranes of the thylakoids. So like mitochondria, chloroplasts also have their own DNA which can synthesize their own proteins because by using prokaryotic like ribosomes. Okay, and they can synthesize not all, but many of their own proteins, and they also replicate through division similar to prokaryotic cell division. So that basically gives rise to the endosymbiont theory, where people say that the eukaryotic cells might have engulfed bacteria, aerobic bacteria, or some kind of bacteria, and they finally have the course of evolution has become mitochondria and chloroplasts. Okay, so basically we have talked about all the cell organelles. We'll stop here today. Is anybody joining? Yeah. So the question that we started with was which of the organelles is basically not membrane bound? That will be the ribosome. Ribosome is not membrane bound. All the other organelles in our question are membrane bound. We have talked about the endomembrane system. We have talked about all the other organelles as well. Okay. So we'll stop here today. It's 8 almost. So does anybody have any doubts regarding the questions that we discussed today? So anybody has any question? Yeah, so there's a question, ma'am. If oils are overheated, can trans fats form in them? Okay, so heating or frying or using any kind of edible oils induces chemical changes, and that can actually create help in the formation of trans fatty acids. For example, if you heat a polyunsaturated oil to a very high temperature for a very long period of time, before it begins to trans isomerize into trans fats, and even then only a very small percentage is converted. So the amount of trans fat generated during heating and frying is still low when compared with the amount of trans fat in partially hydrogenated oils. So yeah, if that answers your question. Sorry. Does anybody have any questions, or we can conclude the session today and we'll meet again next Monday at 6 p.m. Okay. Since nobody is saying anything, I am assuming nobody has any question. So we can meet next Monday at 6 p.m. Thank you so much for joining. Thank you. Thank you. Thank you. Thank you. So I'll end the call for everyone. We'll join next Monday.